Hi guys, so this is Introduction to Bio, and today we're going to be reviewing the cell cycle. So every living thing, every living and dividing cells go through a phase to grow and divide called the cell cycle, and it's like a life cycle for cells. So in the cell cycle, there are 5 stages called G1 phase, S phase, G2 phase, mitosis, and cytokinesis. So there is a G1 phase, G2 phase, S phase, G2 phase, mitosis, and cytokinesis. The G1, S, and the G2 phase together is called a interphase. And the mitosis and cytokinesis together is called the end phase. So let's talk more about each phases more in depth, starting with the interphase. So the cell spends 90% of their time in interphase, and if you see in the previous slide before, you can see that that it spends 90% of its time in this phase called interphase. So in interphase, interphase is where cells get to grow and have its DNA replicated and develop cell functions. So in an interphase, it is important for cells to be able to check all these 3 boxes in order to move on to the next step, or so-called the end phase. So in the first state of interphase is called G1 phase right here. And in here, the newly formed cells increase in size and copies organelles like mitochondria. Through G1 phase copies organelles of the cell, it is important to note that it does not replicate DNA. So that's in another phase. So in the first G1 phase, it increases in size. And it also copies organelles. After the cell undergoes the G1 phase, it goes through this thing called the G1 checkpoint. So in here, the checkpoint makes sure that all the cell is appropriately replicated and checks that the cell is healthy. And if it does not meet the G1 checkpoint's requirements, the cell will go to the G0 phase, where the cell either dies or is in a resting state. So it checks that the cell is healthy. Next, the cell goes on to the S phase right here. And S stands for synthesis of DNA. This is where the DNA is synthesized through the process of replication. So we can write that DNA is synthesized through process of replication. Same as the G1 phase before, the S phase also has a checkpoint that monitors for any replication errors within the DNA synthesis. So the checkpoint is called the S checkpoint. And this is where DNA synthesis is monitored for errors. Now, the final phase of the interphase is called the G2 phase, right over here. Um, in this stage, the cell grows more in size and makes more protein. But the most important job of this stage is to get the cell prepared for mitosis. So it grows more in size, and prepares for mitosis. And of course, the cell goes into a G2 checkpoint as well. This is where it has the final check to make sure all the chromosomes are fully replicated and has no damage. So, all chromosomes are fully replicated with no damage. And this completes the interphase. Now, the cell is ready to go into the end phase if it goes through all the checkpoints. After interphase is completed, the cell should have growth, DNA replication, and have all of its cell functions. So now it's ready to go through the end phase. So mitosis is a type of cell division where it goes through 4 stages to duplicate their DNA and all of its parts. So in other words, this is where the cell actually divides and multiplies. Though mitosis divides and duplicates cells, it does not make sperm or egg cells. It's very important to note that mitosis does not make gametes or sperm and egg cells. So there are 4 stages of mitosis, and I like to memorize it as I like to memorize it as PMAT. So P for prophase. Oh, sorry. So P for prophase, N for metaphase, A for anaphase, and T for telophase. So the first state of mitosis is prophase. Prophase means before, so that's how I memorized that this is the first phase. In prophase, the nuclear membrane starts disappearing and the chromatin condenses and becomes more visible. So if you see on the diagram on the right, you can see that the nuclear membrane is kind of disappearing, and so that the chromosomes can condense. And the spindle fibers start to form and starts extending from one centrosome to another, and the centrioles also move to the opposite poles of the cell. So prophase is kind of a busy phase where it preps the cell to divide. So prophase, one thing it does is chromatin becomes visible, and another one is that spindle fibers form. And lastly, centrioles move to the poles. Then, after prophase is done, the cell moves on to metaphase. I like to think of M as middle because the chromosomes will start to line up in a single file in the middle of the cell. If you look at the diagram on the right again, you can see that the chromosomes are lined up in a single file over here, and the spindle fibers are started to will run from the centrosomes to the centromeres of the chromosomes. If you see, this is spindle fibers, and it's attaching to the centromeres of the chromosomes. So, in metaphase, we can write that chromosomes lines up in the middle. Okay, next, the cell goes through anaphase. A for apart because in this phase, the centromeres of each chromosomes separate, and the spindle fibers pull the sister chromosomes apart from each other, and this will result in a chromatid. And looking at the diagram on the right again, you can see that the chromosomes are separated from each other, leaving with a single chromatid. So in anaphase, you can write that centromeres of each chromosome separates. Right? So lastly, the cell goes through telophase, and this is basically the opposite of prophase, where the chromosomes are now clustered at the opposite ends of the cell. They're now at the opposite end of the cell, and the nuclear membrane starts to reform, and the coiled chromosomes also start to unravel and return to thread-like strands. So in here, we can note that telophase nuclear membrane reforms, and the chromosomes return to thread-like strands. Now, after the telophase is done, this completes the last stage of mitosis. Now, the last state of the cell cycle is called the cytokinesis. If you look back here, okay, so now the last state of the cell cycle is called cytokinesis, and this completes the full process of the cell cycle. So cytokinesis divides the cytoplasm of the cell, dividing the cell completely and making the two cells identical. So the definition of cytokinesis can be written as divides the cytoplasm of cell. So cytokinesis is kind of different because it depends on whether the cell is an animal cell or a plant cell. So let's look at the differences between them. In an animal cell, a cleavage furrow forms in the middle of the cell as cytoplasm pinches inward and the two daughter cells separate from each other. So one of the unique features that animal cells have in the cytokinesis is that cleavage furrow forms. Oh, I missed the A. Okay. Cleavage forms over there in the animal cell. And the two daughter cells separate. So to recap, in an animal cell, the cleavage forms in the middle of the cell, and the cytoplasm pinches inward, and the two daughter cells separate. Now, let's look at the difference between an animal cell and a plant cell. So, in a plant cell, there's a cell plate that forms down in the middle of the cell, and the cell does not separate, and they're held adjacent to one another by a sticky substance called the middle lamella. So the difference between them is that in a plant cell, a cell plate forms. And the two daughter cells do not separate. Instead, the lamella holds the two cells adjacent to another. So the daughter cells do not separate. And it also has the sticky substance called middle lamella. Ooh, I spelled that wrong. Okay, so that completes the full cycle of the cell cycle. But then, as I mentioned before, there is a form of cell division where instead of body cells, sperm and egg cells are produced. So remember how in mitosis we noted that mitosis does not make gametes, or so-called sperm cells and egg cells. So there's another cell division called meiosis, and this is where the sperm and egg cells are produced. Meiosis is carried out in two rounds, meiosis 1 and meiosis 2, and it's very similar to mitosis' 4 stages of PMAT, and meiosis also follows the outline starting from prophase 1. So let's write prophase. So here is when the chromosomes condense, and this phase is really important because there is an important process called synapsis and crossing over that occurs here. And during a synapsis, chromosomes pair up with homologous pairs, or so-called like chromosomes that are approximately the same size and contains the same type of genes. So that the crossing over can occur. So chromosomes pair up with similar chromosomes that have the same size and same genes, so that they can transfer genetic material. So you can write here is that synapsis occurs and crossing over occurs. Crossing over is a process in which homologous chromatids exchange genetic material to ensure a greater variety in the gametes. And then that is prophase 1. Now, going on to metaphase 1, it is a little different from mitosis because the chromosomes are going to be in pairs instead of single line along with the middle of the cell. So, metaphase lines up in the middle. Now going on to anaphase 1. Similar to mitosis before, the homologous chromosomes are going to separate. So we can write that in anaphase 1, you could say homologous chromosomes separate. Now, final stage of meiosis 1 is telophase. In here, the cells would now have 23 chromosomes, but still 46 chromatids. This is why we need to go through this process of meiosis again, because we need 23 chromatids for each. But then after completing meiosis 1, we have 23 chromosomes, but we have 46 chromatids. In order to make this number separate into 23, we need to go through the meiosis once again. So it's actually, meiosis 2 is exactly the same. Everything's the same except that in prophase 2, there is no crossing over. So no exchange of genetic material. It only happens in prophase 1. Now, in metaphase 2, the chromosomes line up in the middle, but it is in a single file. It's not in the middle, the two chromosomes anymore. It's in a single file. So we could say that lines up in a single file. Oops, pen is bugging out. Single file. Okay, so next in anaphase 2, the chromatids are going to be pulled away instead of the chromosomes being pulled away. So we could say that chromatids are pulled away. And finally, the last step is telophase. Telophase 2. In here, now there will be 4 new cells with 23 chromatids in each cell. So that basically completes everything from the cell cycle. So if we take the summary of this, is that G1 and G2 are gap phases starting from, So if we review everything in total, so in the cell cycle, there are 5 stages: G1 phase, S phase, G2 phase, mitosis, and cytokinesis. The G1 phase, S phase, and the G2 phase make up interphase, where the cell basically spends most of its time. If you see over here, you can basically practically tell that it lives here. And then it goes down to this little section of end phase, which includes mitosis and cytokinesis. So in an interface, again, it is where the cell has growth, has DNA replication, and has developed cell functions. And after all that checkpoint is met, all the requirements are met, we go on to mitosis. And this is where the cell actually divides and duplicates. And if mitosis also has four stages or phases called PMAT. So in the prophase, this is where the chromatin becomes visible, the spindle fibers form, and centrioles move to the poles. Good diagram here. And then the metaphase is, remember, it lines up in the middle. The chromosomes lines up in the middle like this in a single file. And then the anaphase is where the centromeres of chromosomes separate. So after it's lined up in a single file, the spindle fibers pull away the chromosomes from each other, making it chromatids. And then finally in the telophase, the nuclear membrane reforms and the chromosomes return to thread-like strands right here, basically the opposite of prophase. And then finally we have cytokinesis. The definition of cytokinesis is that it divides the cytoplasm of the cell, basically completing the cell division. And it depends on either if it's an animal cell or a plant cell. In an animal cell, a cleavage forms. In a plant cell, a cell plate forms. And in an animal cell, the two daughters separate completely, if you see in the diagram here. But then in the plant cell, the middle lamella holds the cells together like this, so it does not separate. And finally, this is where the sperm cells and the egg cells get to divide because it doesn't divide in mitosis, and it follows two stages, meiosis 1 and meiosis 2, and it also follows PMAT, but it's kind of different because in prophase 1, there's this thing called synapsis and crossing over where it exchanges genetic material, and everything else is practically the same from that. But then in meiosis 2, the difference is that in prophase 2, there's no crossing over, there's no genetic exchange, and from that, everything else is the same, resulting in 4 new cells with 23 chromatids each. So that's like the overview of the cell cycle. If you guys have any questions or what I should review next, please comment down below. Thank you.